All right, folks, let's get started. Should we start with a sound check? Can you hear me in the back row? Can you hear me in the back row there? Excellent. Wonderful. Before I get further into the biochemistry, which is always more fun, the lecture from Wednesday is not yet up on Google. I have made a couple of attempts to get it on Google. So far, both times Google has burped and said no. I'll try again, but I just want to let you know I am trying. You don't need to write. It will be there when I can get it there. Hopefully, if that doesn't work at some point, I'll probably try YouTube instead, but I think YouTube has a time limit, so we'll see. Any questions from Wednesday before we go on? Okay. Now, we talked on Wednesday about making RNAs, about the polymerase that can do that, how it has a hand, thumb, and palm, kind of like DNA polymerase. And we talked about what it takes to turn on genes at the right time and how it's a little different between prokaryotes and eukaryotes. But making an RNA is not enough. What we see in the cell is that RNAs are extensively modified and that those modifications are very important to their function and to their lifetime. And so part of what we'll be talking about today is RNA processing. And what I'm starting to talk about right now is actually RNA processing in eukaryotes. There is a little bit of fuzziness among the prokaryotes in that there's some differences about how they handle it across different branches of the prokaryotes. What I'm saying today applies to all eukaryotes. And in eukaryotes, all RNAs are modified. This modification actually starts during transcription, before the RNA polymerase has finished its job. And to the extent that there are some of these processing enzymes that sit on that charged tail, the C-terminal domain of the polymerase, so they're right there ready to do things. At the 5' end, there are several interesting things that happen. There are 3 activities that are needed to make what's called the 5' cap. There's a triphosphatase that can cut off the triphosphate tail. A guanylyl, I have to fix my thing, don't I? Pointer options, no options. A guanylyl transferase that can put a guanine residue on, and a methyltransferase that can then methylate that. And the end result of that is what's called the 5' methyl G cap. And if you look at it, the odd thing about it is that it's a way of giving a messenger RNA 2 3' ends. So what originally looked like the 5' end, with these modifications, now looks like a 3' end. And there's the methyl on the G that modifies that base. And so this 5' methyl G cap ends up really modifying the availability of messenger RNA and how it functions in the cell. And it has to be important for the cell to invest that many different enzyme activities into generating it. At the other end of the messenger RNA, at the 3' end, there's a process called 3' adenylation. And what that consists of is recognizing the sequence AAU, AAA, cleaving it at that spot, and then adding adenine residues, lots and lots of adenine residues. 20 to 50 is not uncommon. This is an enzyme that does not need a template. The only thing it can add is A's, and so it just extends it that way. The fundamental reaction is the same as an RNA polymerase. But this poly A tail, as it's called, turns out to be a signal for a number of different processes. And one of them is it's a binding site for something called poly A binding protein. And poly A binding protein can bind at the poly A and, in effect, cover up one of its naked ends. And in doing that, it tends to increase the lifetime of the messenger RNA. And so the presence of a long poly A tail can change both the lifetime of the message, how long it exists in the cell, its half-life. It can also affect how well it's translated. So of 10 different messages, if one of them has a better poly A tail, it's going to have a better odds of being translated. I first ran into poly A tails when I was a postdoc, not familiar with molecular biology, and innocently asked in our lab group, so what's the function of the poly A tail? And at that point, the only thing they were used for was to isolate them on poly U columns. And someone in the group just said, God put him there so he could isolate messenger RNA. It was a good another 10 years or so before an actual function for them was discovered. There are some messenger RNAs that have no poly A tail. And these tend to be messages that are short-lived, so that by having structural features that make them not targets for the polyadenylation, the cell has a way of having a population of rapidly turning over messages, which gives them more rapid control of the up and the down of it. So that, for instance, some of the early response genes for viruses and things, and some cell cycle genes have no poly A tails. This is just a reminder that I started off with the last time, that a week from today there will be the 4th exam, and Monday the 7th will be the final exam. The last hour exam does include all the way through the end of chapter 20. Are there any other questions about the course matters beyond what I have here? Okay. We talked before, a little earlier on Wednesday, about some DNA binding proteins that had roles to play with regulating genes bound at the major groove. These are some what could be called architectural proteins to a greater extent. So for example, here is the TATA binding protein modeled in contact. Here is another one. SRI is involved with sex determination. HMG stands for the high mobility group. They're the big proteins that are involved in chromosome structure. This is integration host factor. It's the protein that's needed to have a virus integrate into the host. These are all proteins that are more sort of the architecture of the DNA. And interestingly, they all bind at the minor groove and end up changing the overall conformation of the DNA. We've talked about initiating transcription and how the polymerase decides where and when to start. We talked a bit about elongation, that the polymerase needs to be phosphorylated in specific ways to permit continuing elongation. So there's a beginning, there's a middle, there's an end. There has to be an end to things. And so to terminate RNA synthesis, something needs to happen. Just simply pausing is not necessarily enough. An RNA polymerase can pause, hold on to the transcript, stay on the DNA, and wait for something else to happen. So pausing by itself is not enough to end transcription. In prokaryotes, one of the features that seems to be involved with terminating transcription, actually making the polymerase release its RNA and get off of the DNA, are hairpins. So a GC-rich hairpin would be a sequence that has a lot of G's and C's that can make a little local secondary structure that's different. It's just a little kind of a pin like that, followed by U's. And so you can imagine as the polymerase is going along that it encounters that hairpin, and that is part of the signal. In eukaryotes, it's still more of an open question. And I throw this in partly as a way to show that there are things we still don't know about biochemistry. Apparently, part of the cleavage polyadenylation factor, the one that adds the poly A tails, plays a role in termination in some extent. There are cell cycle associated proteins that may contribute, but it's still somewhat of an open question. In Saccharomyces cerevisiae, so this is the yeast that gets used to make beer, there are specific sequences for promoters where it starts and specific terminator regions, and they are in physical contact throughout the entire time, before transcription starts to the end of the release of that message. And so that means that this gene is sort of a loop that the polymerase can go around and release its product, can go around again, and so on. So it creates a little, very local domain for that gene product. So eukaryotic genes have methyl G caps. They have poly A on their 3' end. Most eukaryotic genes, although not all, most of them have introns. And intron stands for intervening sequence. So it's a little bit or a lot of RNA in the transcript that is not going to be there in the final product. So it's intervening between the coding sequences. In order to have a mature, functional transcript, those introns have to be processed out and removed. The exact frequency of these various introns varies with the complexity of the organism. So here you can see in yeast that the most typical number is 1. They're, of course, a very small genome and a small cell with a fairly limited variety of things that it does. Here are fruit flies, where the average numbers are more around 2 to 3, but there are some large numbers. Here we are in the 5 to 10 range, a few up even higher. And in the mammals, clearly, on average, there's quite a bit more introns per gene. Very few that have only 1, and some up here having in the range of 30 to 40. The largest protein in our muscles is 1 million molecular weight as a finished product, but its transcript is larger still and has at least as much intervening sequence to be taken out as it has actual coding sequence. So these introns can be a very massive part of the transcript. And the removal of introns is an interesting sort of dicey project. The process is called splicing. If you've ever had to splice parts of a rope together, you can use that as sort of a metaphor for this. So somehow or other you're going to have to cut the RNA and then splice it back together with that intron taken out. And some of the common features that we see in introns are features at the 5' end of the intron, where there's a sequence like that, and the 3' end of the intron sequence like that. And then partway in, it's 20 to 50 bases, so it's obviously not a real precise thing, there will be an adenine that is going to be used as a branch point. And you'll see in a second what I mean by a branch point. The reaction looks something like this. So in the first step of the splicing reaction, the G here is going to undergo a transesterification. So it's a phosphodiester to begin with, because it's an RNA molecule, which is phosphodiester bonds. So it's going to be transesterified. It's going to transfer the ester linkage from the 5' end of this over to that A, that is the branch point. And it's going to esterify onto the 2' OH. Tells you right there that DNA couldn't do this. DNA doesn't have a 2' OH, so DNA couldn't possibly be spliced. That leaves a free 3' end at the leading part of it, and a branched piece of RNA as an intervening step. This new free 3' end can attack at the other side of the splice point in another transesterification reaction and leave us now with an intact piece of RNA from which the intron has been completely removed. And that intervening sequence then is just going to diffuse away and be degraded. This intermediate is sometimes called the lariat intermediate, and you can see why. It's like those lariats that the cowboys like to use. Well, why bother? Why have introns? And there may be several reasons, but certainly one reason is that by having introns, a fairly small number of genes can give a much larger variety of products. And this may be a little bit clearer if we look at a specific example. So here is the gene here for one of the muscle proteins. I think this is tropomyosin, but I'm pretty sure this is tropomyosin. And this is the gene with the different segments in it. So the parts that are white here are introns, And the parts that have one color or another are going to be exons or expressed regions or actually translated into protein. So the introns are taken out, leaving behind the exons as one continuous mRNA. And when you look at what happens, you see that there's sort of a mix and match approach to the structure of this protein. So all of them have these sequences here at the beginning. And all of them have these sequences here in the middle, or here. But in striated muscle, and striated muscle is like the skeletal muscle in your arms or your legs or your eyeballs, it has these unique purple sequences there and there. And by biochemical experiments, you can probe and ask, what do those sequences contribute to the function of that protein, specifically in striated muscles? Smooth muscles are, again, muscles are going to have a lot of contractile function, but they are much less highly organized than striated muscles. They're under the autonomic nervous system, not under the voluntary system. Smooth muscle is the kind of thing that makes your guts work or makes your blood vessels keep tone and so on. And smooth muscles have here the sort of peach-colored thing here, a salmon-colored thing, and a long salmon-colored tail. In the fibroblasts or other non-muscle cells, so the cells that go around muscles, adipocytes, skin cells, things like that, they have the sort of the basic stuff. They have these features that the skeletal and smooth muscle had, and they have the longer chunk that was present in the smooth muscle. And then the brain form is the simplest of them all. It has only kind of the minimal package, if you like, of what it takes to make this protein work. And it's a matter of considerable interest to people to look and see why that patch of sequence allows that protein to function in the context of striated muscles and so on. And this general theme of having one gene with a number of different exons that can be spliced differentially is repeated in many contexts. This is one of the major ways, not the only, but one of the major ways that tissue-specific proteins are generated. And you may remember we talked about tissue-specific enzymes for glycolysis and gluconeogenesis and so on. And this is one of the ways that's accomplished. There are several different kinds of introns, depending on how they're processed and so on. In type 1 introns, they are self-splicing. The sequence of the RNA is sufficient to provide for that splicing process. And we see some type 1 introns, the self-splicing ones, in nuclear mitochondrial chloroplast genes, if they code for ribosomal RNAs, messenger RNAs in those contexts, and tRNAs. This kind of self-splicing intron is also seen in the group called the archaea, or some of them sometimes called the methanogens, although there are methane-forming bacteria elsewhere. So this is one of the features that makes the archaea more like eukaryotes than like other prokaryotes. A second class of introns is also self-splicing, but it's not found in the nucleus, only in mitochondrial and chloroplast genes, in a wide variety of things. Type 3 introns are only found in eukaryotes. This is uniquely a eukaryotic trait and is not shared with the archaea. And this is going to involve using SNRPs. I always like that name. Small nuclear ribonucleoproteins. So SNRNPs or SNRPs are going to assist the process of splicing in a structure called a spliceosome. So while type 1 and type 2 have the sequence in their RNAs and can splice themselves just by their intrinsic properties, type 3 introns are going to need assistance from the SNRPs. The type 1 and type 2s are so sufficient at self-splicing that you can make those RNAs in vitro as pure RNA and watch them splice themselves. Whereas by contrast, type 3 really needs these SNRPs. The fourth kind of intron, type 4 introns, are in transfer RNAs. And this splicing process actually requires energy and an endonuclease. And interestingly enough, this one is also present in the archaea. So we see 3 of the 4 types are present in the archaea. It's only the type 3 type with the SNRPs that's uniquely eukaryotic. Any questions there before I discuss in a little more detail. Okay. This is sort of a graphical representation of how those things work. So here's a group 1 intron, and you start by having the transesterification reaction coming in. So now you have sort of a dangling end with a second attack. You have final closure, and you have the free piece of RNA that got cut out. Group 2 looks more like the diagram I showed you initially, where you have the transesterification reaction over to the A with a lariat kind of thing as an intermediate. The new 3' end can come in, transesterify, close up, so you have an intact RNA again, and you have a free floating lariat. For a little while, it will be degraded. In pre-mRNAs that need SNRPs, the RNA is doing the same kind of thing as in type 2, but it will require the assistance of the SNRP proteins to do it right. If you think about that sequence I just showed you with so many introns and exons, you can imagine you might want some machinery to help you to pick the right introns, the right exons to do, right? If in some tissues you're going to use this set, and some tissues you're going to use that set, it's not too surprising. It takes more than just self-splicing to make that work appropriately. And then in tRNA, you have sort of the nascent tRNA, if you like, or the initial transcript. Part of that has to be cut by an endonuclease, and because it's cut free, then there's a kinase and a ligase that ties it back together at the expense of some ATP. This is a diagram of how the spliceosomes work. And you can see here is the initial transcript. Sorry, this is the DNA that's being transcribed. Here's the RNA polymerase II, and here's the growing messenger RNA coming out here. This part is an exon, and there's the 5' splice site where things need to start happening. And the U1 SNRP will bind at that spot. The RNA polymerase goes on moving. It's still in contact with the transcript. So this is all happening during transcription. Okay? This kind of splicing can happen during transcription. The second SNRP to come in is the U2 SNRP that can bind at the branch site. Interestingly enough, these are called snRNPs. So it's a ribonucleoprotein. These are proteins that have RNA bound to them, and the RNA is an essential part of how they function. So these are not simply proteins that help with splicing. These are ribonucleoproteins, where both the RNA and the protein components are essential for the splicing to occur. Then in this diagram, let's see if we can find U5 here. Here's U2, here's U4, 5, 6. Here we go. U5. Here's U5. U4 and U6 come in and help finish assembling this spliceosome. And you can see that all of these things have RNA molecules sort of hanging off them. So, this is showing how the RNAs of the small nuclear RNAs play a role at helping to guide these contacts to be made. And I don't expect you to memorize which contacts are being made where. The point I'd like you to pull out of this is that the ability for these RNA components of the SNRPs to bind to parts of the sequence is what allows it to select one part of the gene to be spliced together, one part of the transcript to be spliced together, and not others. But you can see here there's a U5 snRNA and U6 snRNA that are actually making base pairs to some of the transcript. And I think you've heard me say before that degradation is as important as synthesis, that controlling breakdown is always important as the making of something, and that there's an off switch whenever there's an on switch, and that kind of thing. And this is just as true for messenger RNA. It was not always appreciated. During most of my early scientific career, people never even thought about mRNA degradation. If they had realized it, they would have realized that they should have imagined a world that was filling up to their eyeballs in RNA, because if it's never degraded, it's going to just accumulate. So obviously degradation was happening, it just wasn't being paid attention to. At steady state levels of RNA are a function of the synthesis rate and the degradation rate. And so you can either have a high rate of synthesis and a high rate of degradation and get X amount, or a low rate of synthesis and a low rate of degradation and maybe have the same amount, right? So it's the balance of the two that's going to determine the steady state, and whether you do it fast or slow is probably going to reflect whether you need to change it quickly or slowly. If it's something that you're usually going to have around all the time, it's usually going to have a long-lived messenger RNA. If it's something you need to turn on and off, like the messenger RNA for the cyclins that Carissa talked about, usually the synthesis will be fast, but so will the degradation. So that gives you a spectrum to control things over. And how do we do that? Well, we mentioned that the modifications on a eukaryotic message include the 5' methyl G cap that makes it look as though it has a 3' end, but it is the 5' end of the message. A 3' poly A tail that was added. One of the early things that happens before a message is degraded is to lose that poly A tail. And there is a deadenylase that can eat that poly A tail off. That will make the message more likely to be a target for a decapping enzyme, which would cut off specifically the cap, just the cap. And then when you have an RNA that is naked at both ends, it has no poly A tail, it has no methyl cap, there are exonucleases that are available that can then nibble in from both ends and degrade the rest of that message. So you can see there's a significant amount of machinery at degrading it as well. Questions? Okay. In recent years, we've come to appreciate that there are ways in which RNAs can control other RNAs. And siRNA is called that for small interfering RNA. It's the RNA behind a phenomenon called RNA interference, where one gene can interfere with another one. And it's turning out to have a role both in protecting cells from RNA viruses, but also plays a role in development. It's not just to protect cells against pathogens. And what's involved? Small interfering RNAs work something like this. The parent molecule is one that will be double-stranded, and that is a substrate for an enzyme called dicer. And dicer chops it up into smaller pieces. So these would be the small interfering RNAs. They're not very big RNAs. And these small interfering RNAs can then interfere with an mRNA that can be hybridized to. So the small interfering RNAs are sequence specific. And if they form complexes, there are RNases that will attack only at the places where the small interfering RNAs have made a complex. And then once you start nicking up a messenger RNA, the rest of it can go pretty quickly. If you nibble at two places on that messenger RNA, you've created bare ends. And those exonucleases I talked about can eat the rest of that up. So this is a way of, on the one hand, protecting against double-stranded RNAs, because the small interfering RNA can interfere with that. But we're also discovering a number of places in development where important genes are turned off by synthesis of a small interfering RNA by the cell. This is showing you a little bit more detail on how siRNA works. Here is a synthetic siRNA that's been made for the particular target. And you have to make a RNA protein complex, the RISC complex, before it can actually interact with its target. And so this RNA protein complex is the one that can actually then interact with the mRNA, and you can see here some hybridization occurring while it holds on to the other strand. The nuclease can attack. This complex is still intact and is still able to go back and attack something else, whereas the mRNA that's been nibbled in two places is going to get completely degraded. So this is a catalytic way to get rid of RNAs. Since one RISC complex can attack multiple messenger RNAs, it's catalytic and it's very efficient. And to give you a feeling for just how efficient, this is an example of a cell in which the target protein is on, and one in which it's been turned off by siRNA. So this allows a greater depression in protein levels than many other methods do. Some people are hopeful that perhaps it might someday be able to be used as some kind of a gene therapy. So we'll see. That's good ways in the future. This is sort of showing you, a yet closer look at all of this. So in order to see how you can make the attack, the small interfering RNA has to unwind a bit, and you create this complex. Let's see, where do I want to walk this? Okay. You can have two different kinds of interference. You can have one where you have a perfect match. So if you have a perfect match, you can then be a target for the nucleases where you've cleaved it, and then that will be degraded. If you have a mismatch like this, where some but not all the bases stick, the RISC complex will simply stay there and block further translation. So when it's a perfect match, it's a catalytic inhibitor and can degrade multiple copies. When it's an imperfect match, it's a one-for-one silencing. It's not going to be able to, you have to have one siRNA for every message. This is sort of a more dramatic way of looking at the dicer complex of how it can cut up RNAs into smaller pieces. And what they're suggesting here is that perhaps rather than depending on dicer to cut up transcripts from the nucleus, possibly we can feed small interfering RNAs into the cell by packaging them in liposomes. And so it'd be interesting to see whether or not that ends up being a useful approach or not. Okay. We can see this working in an animal. This is some fairly recent work. So the ApoB genes are genes that are involved in LDLs. Remember, the LDLs are the bad cholesterol. They're the low-density lipoproteins that add cholesterol to your veins. And in some humans, they have high levels of ApoB, and so they have high levels of LDL. And the question they're asking is, could we reduce the levels of LDL by using small interfering RNA to turn it off? And the idea here was to covalently couple small interfering RNAs for ApoB to cholesterol molecules. And so that's kind of a cute trick, right? The cholesterol will take it where cholesterol goes, and that helps take it up as well as to stabilize it. The good news is it can, in fact, turn off LDL. So you can see here decreased levels of LDL and of chylomicrons. Unfortunately, only 1 out of 84 mice responded. So it's not yet to the point where we can use this on a routine basis for everybody. And also, mice don't always have exactly the same biochemistry as the rest of us. As my husband once said, I can tell you how to cure cancer in a mouse, but it takes a little while to get from mice to people. What we've been talking about so far has been the processing of messenger RNAs, and those are the most diverse RNAs, of course, because you need a different mRNA for every protein. But mRNAs are not the most abundant RNA. The most abundant RNA are the ribosomal RNAs, because you need ribosomes to translate those messages into things. And you need a lot of them. And so there's a separate polymerase, the Pol-1. Polymerase 1 is the polymerase that specifically makes the ribosomal RNAs. There's a specific structure in the nucleus called the nucleolus, or nucleolus, depending on who you learned it from, meaning the little nucleus. It's a little dense spot inside the nucleus that's specialized for ribosome transcription and synthesis and assembly. It is, as I said, a very abundant RNA by comparison to the others. The initial transcript is going to be of a particular size that's characteristic. Why is it buzzing at me? A particular size that will be characteristic for this particular species. They're showing you here a 45S RNA. That's a good ballpark number for the initial transcript. S is for Svedberg's. It's a measure of how fast things sediment, how fast they'll come down in a centrifuge tube. It doesn't exactly map to mass, but it's a measure of relative size. So if we start with an initial 45S ribosomal RNA transcript, it's going to have to get cleaved in a number of places before we start to get out of it the RNAs we need. And so we're going to throw away part of the 5' end, we're going to chip off 2 pieces in the middle, and that gives us an 18S, a 5.8S, and a 28S ribosomal RNAs. So the large, the small, and the 5.S RNAs. These are pretty typical sizes for ribosomal RNAs. There are some variations with species. These RNAs are also going to be extensively modified. This is pseudouridine. So you can take an ordinary uridine and modify it by like that. You can methylate different bases. And you might well ask, if you've got this huge, and 28S is huge, trust me on that one, RNA, how do you pick out where you're going to modify it? This is through another group of small RNAs. So this is small nucleolar RNAs. So these are, again, small RNAs, but these are usually found in the nucleolus, and they assist with targeting these modifying enzymes to the correct place. These RNAs themselves are not enough to make a functional ribosome. There needs to be a lot of ribosomal proteins. So a typical eukaryotic ribosome will have another 80 ribosomal proteins that interact with these 3 RNAs. And when we talk a little bit more about our protein synthesis, we'll talk a little bit more about the structure of the ribosome and how it works the way it does. But you can see there's really quite a lot of machinery needed to produce a ribosome. Another RNA that's very important to life are transfer RNAs, tRNAs. We're going to look in more detail at how tRNAs match up the sequence of the messenger RNA to the particular amino acid that needs to be inserted. They're made by a different polymerase, by polymerase 3. tRNAs are also pretty abundant in the cytoplasm by comparison to messenger RNAs. We talked about some of how its introns are processed, but there's also some trimming and cleavage that goes on. RNase P is an interesting RNase. So this is a ribonuclease. It can cut RNA, but its ribonuclease P is one in which the catalytic residues are RNAs. The protein part is there to hold the RNA to do the catalysis. But in this case, it is the RNA that is the catalytic residue. tRNAs are very heavily modified. About a quarter of the bases have pseudouridines, methyl groups, and other kinds of things. I've mentioned about how splicing can occur for tRNAs. They are not all spliced. Some are spliced, some self-spliced. Depends on the different tRNA in particular. These are some of the other modifications. I don't expect you to memorize them, but just to give you a feeling for the kind of zoo of bases that they have. Here's an isopentenyl adenosine. Here's a methyl adenosine. Here's a methyl cytidine. Here's a dimethyl guanosine, dihydrouracil. So a lot of modifications. And then eukaryotes add a CCA piece at the end of the tRNA. That's not coded in as part of the transcript. It's added on post-translationally. And this is probably as good a time as any to talk about RNAs as enzymes. If you think about ribonucleic acid and you compare it to protein, which we know about as an enzyme, there are some very important features that they have in common. So both of them have a flexible linear backbone. RNAs can undergo non-standard pairing. So these are some of the non-standard pairings that are possible. These are all permissible pairings that provide for hydrogen bonding other than Watson-Crick base pairs. But these are all conceivable or permissible pairings. Taking those two together, the flexible backbone and the non-standard pairing, you can get tertiary structure. If you think about the tRNA I've been talking about, it folds up into a very particular shape. That's tertiary structure. So the double helical structure that we're familiar with DNA and with double-stranded RNA can be thought of as secondary structure. But when you add in these other features, you can end up with a very specific packing in space that really can be called tertiary structure. And those tertiary structures can include the capacity to bind metal, for instance. So between the residues of the nucleic acid itself and the metal that can bind to some of these, you recognize that there's the potential there to be a catalyst, to be something that can undergo reactions in a specific way again and again. So this is something that was not appreciated about RNA until fairly recently. RNA was thought of as just kind of the tape you feed into the tape player to get music out. The first ribozyme, as it's called, so a ribozyme is a term for an RNA that can make a reaction happen. The first ribozyme was a plant virus that could cleave itself. So it was transcribed as a big piece and it cleaved itself into the smaller pieces. I am old-fashioned and do not consider it really a catalyst because it used itself up in the reaction. It was a one-hit wonder. But we do know true RNA catalysts now that can undergo multiple rounds of catalysis where they are not used up in the reaction. They can do it more than once. This shows you some of the structure, and this is a self-splicing RNA showing you that. So what I'm going to move into now is not in the textbook. It's sort of an extension of the various things we've been talking about. I may not be able to get to the end of it, and whatever I get through today is as far as I would expect you to know. If people want to stay to hear the rest of it, you're welcome to do that. But what it is it's building on some of these things that we've learned about RNA to look at the question of what's been called the RNA world. A number of people, actually for a couple of decades now, have been suggesting that before life was the way we see it, there was a time in which it was simpler and didn't use DNA, it had RNA instead as the thing. And the reason for raising that possibility is because cells as we know them right now are so complicated. And the RNA world was proposed as a way to have something simpler than a cell today that still had a visible connection to where we are now. If you think about the world as we know it now, you've seen this diagram before, I don't know if you remember it. We have interlocking systems. So these arrows here show how information can flow. So the sequence of DNA determines the sequence of more DNA in replication. Carissa talked about that. The sequence of DNA determines the sequence of RNA. That's what I've been talking about. The sequence of RNA determines protein. We'll be talking about that in chapter 20. So that's the central dogma. Information flows from DNA to DNA, from DNA to RNA, from RNA to protein, most of the time. Small exceptions. But these reactions themselves require their own products. That is, for DNA synthesis to happen, you need RNA and you need protein. For RNA synthesis to happen, you need RNA and you need protein. And so you have a system where you need the products of the system to make the system happen. It's an interlocking, self-referential problem, and it's part of the challenge that trying to think of how life could start presents. The other thing cells have to have is some kind of an enclosure. It's not enough to have these parts that can interact with each other, but you need to have the material for it that's appropriate in the space around it. And we achieve that nowadays through a membrane that's semi-permeable. So there has to be some way to enclose your informational system for it to be considered a cell. It has to be a semi-closed. You need to get stuff in and out, but you don't want everything to go all the time. And so that really raises some serious questions if you try to think about an RNA world, a world without DNA, and perhaps even a world without protein. One real problem is how can you get replication without what I'm calling recursion? How can you get replication without needing the products of replication? And a very interesting recent finding has given us a suggestion about that. Other problems in an RNA world: how can RNA last long enough to do anything? Those of you who've done lab work know that RNA goes away if you sneer at it. It is not your favorite stable molecule. And it turns out that there is simple inorganic materials that stabilize RNA that we didn't appreciate until recently. What kind of reactions could RNA catalyze? I mean, you need a spectrum of reactions to make life work, not just one or two. How many can they do? And can it regulate? You know, an informational system that has no capacity for regulation has no capacity to respond appropriately. And so an RNA world in which RNA couldn't regulate itself would be just as dead as without having catalysis. And finally, how could a simple system be bounded? How can you set off a place for it to happen and not everything all at once? And in all of these questions, interesting findings have come up in the last 5 years. And so it's been an extraordinarily fertile time for finding out aspects, both about life as it is now, but also perhaps on what life was like earlier. Let's start with the replication without enzymes. Someone had proposed that it would be nice if there were molecular midwives, molecules that could help replication to happen without themselves being part of it. And this is something that Karen Smith had proposed. This is a brilliant guy whose paper I have up before. Earlier, he said, you can look at an arch like in a church or something, and you can't imagine how it'd be built, but it started off with scaffolding inside the arch, and you finish the arch, and now it's self-supporting. You can take the scaffolding away. And this is an analog of Karen Smith's scaffold. It could have been helpful at the beginning before we had enzymes to catalyze polymerization. And the observation is that proflavin, which is this molecule here, can intercalate between nucleotides and stabilize them in a way that accelerates polymerization. So it can act in a completely non-specific way, independent of the RNA or the DNA that it's polymerizing way to foster polymerization. You can see from this cover that this is quite recent. This was just observed and it was just reported in 2004. And I won't try to go into all the details of it, but this molecule, proflavin, occurs in the absence of life. We see it in molecular clouds around planets. If you look at the shape of it, you can see that proflavin is kind of the same shape as a Watson-Crick base pair, and it's aromatic as heck. And so proflavin has the capacity to do stacking interactions between Watson-Crick base pairs. The presence of proflavin in a mixture like this, so this is a template here, and these are some substrates that they want to see if they can catalyze a bond forming. Adding proflavin to this reaction mixture increased the rate 1,000-fold. So this is a very significant catalysis over the absence of the reaction. And this is showing how that intercalation probably facilitated things. By holding things together for a longer time, it allowed that bond to form. And this is just getting into some of the actual evidence for that. I don't know if you want to look at it in detail or not, but that as you get more proflavin, you get more product. This is showing you that DNA is the most assisted by it, but that RNA has a broader peak. So there's a broader range over which proflavin would be helpful to RNA. And again, that 1,000-fold acceleration. Here's how fast it happens in the absence of the proflavin. Essentially negligible. And then this is the quantitative results, which we can. And I think I'll skip that, and maybe I'll call it the end. But if people want to stay and hear the rest of it, I'll be happy to talk about it. Otherwise, we'll call it a day. Have a good weekend.
All three are RNAs of 5S, so the ribosome 60 units?
There is one of the small RNAs that's made by polypeptide. I believe that's true. But there are two small RNAs in ribosomes, one of which is made by polypeptide. I'm not going to worry about going down to that level of detail.
All right.